Okay, so hello. Welcome to Thursday PM's Group 4 final presentation, where we're looking at the teragenic effects of low ethanol, which is 0.5%, on zebrafish in the nervous and immune systems. Here are all of the authors and as well as their affiliations to the University of New England, the corresponding author and email, as well as a funding statement, and the keywords are ethanol, zebrafish, RT-PCR, immune system, nervous system, and morphology. So just before we get into the actual presentation, here's an overview slide. The purpose of this presentation and this whole project was to determine the effects of ethanol on 4 different target genes in the organism AB house wild type zebrafish. The biological systems were the immune and nervous systems. We are going to go through an introduction of why this is important, go through the experimental methods, results, and discussions, which was RT-PCR, a Western immunoblot, enzyme assay, and morphology. So the overall impact slide, zebrafish were exposed to 0.5% or low dose ethanol media in order to better understand if low doses of ethanol exposures during development can influence expression of target genes in the nervous and immune systems. This research can be used to further understand fetal alcohol syndrome disorder. It is important to continue research on the effects of ethanol exposure during pregnancy on embryonic development to better understand fetal alcohol syndrome disorder and its larger economic and social impacts. So for an introduction, pterogens are substances that can cause harm or abnormalities during pregnancy to a fetus. Ethanol has teragenic effects on fetal development across many species, including humans and zebrafish. Common exposure is through maternal alcohol consumption for humans, which is why it's very prevalent, as it's estimated that 1 out of 13 women drink alcohol during pregnancy, and that FASD impacts up to 5% of children in the United States each year. So a lot of research has been going into this because it is a health concern in humans, and previous study has looked at ethanol exposure in the nervous system. Ethanol can change the expression of certain genes during embryonic development. Previous studies have showed that ethanol exposure can cause cell death within the central nervous system of zebrafish models. And it showed in a 2004 study that it caused a reduction of brain size and expressed neurobehavior effects. Previous studies also saw abnormal brain development from infant human autopsies who were pre-exposed to ethanol during pregnancy, and they found that there was corpus callosum malformations, poorly formed cerebellums, and signs of either hydrocephalus or ventromegaly due to that prenatal ethanol exposure. Another study also found that exposing zebrafish to ethanol had led to smaller eyes, reduced body lengths, spine curvatures, and a decrease in sensory neurons. Since we're also looking at the immune system, we found previous studies that looked at ethanol exposure in the immune system. The immune system is important to fight off bacteria and toxins with the inflammation as a main defense mechanism. Ethanol was found to increase activation or the amount of microglial cells in juvenile zebrafish. This led to a greater inflammation response seen in the 2019 Brown study. This could lead to immune system attacking their own body cells in an autoimmune response. And previous studies also suggested that human babies are 3 times more likely to get infections if their mothers drink heavily while pregnant. So now looking at the purpose, to determine the effects of ethanol on 4 target genes in the immune and nervous systems of zebrafish. And the hypothesis was that if zebrafish are exposed to a solution of 0.5% ethanol during development, then there will be little to no significant change in the expression of target genes.
Okay, so our model organism was AB house wild type strain Danio rerio, which is zebrafish. They are great to use in the lab because they are easy to handle and they develop fast after fertilization. It is hypothesized that humans share 70% of genes with zebrafish. They have similar anatomy, including similar immune and nervous systems. And zebrafish also have sensitivity to environmental toxins. So materials and methods for husbandry and treatment paradigm. So we use the AB house wild type strain zebrafish. They were exposed to ethanol 48 hours post-fertilization. The drug media was a 0.5% media of ethanol that was changed every other day. The larvae were harvested 7 days post-fertilization, and they were euthanized via a 1 to 5 dilution of 2-phenoxyethanol. There was a waiting period of 10 minutes that followed the euthanization of the zebrafish. So for each experimental group, so for RT-PCR, the experimental group 1 had 25 zebrafish, experimental group 2 had 17 zebrafish, control group had 26 zebrafish. For electrophoresis and immunoblot analysis, experimental group 1 had 20 zebrafish, experimental group 2 had 22 zebrafish, and control group had 23. For enzyme assay, experimental group 1 had 15 zebrafish, as well as experimental group 2 and the control. Morphology had 9 zebrafish in experimental group 1, 13 in group 2, and 10 in the control. So the genes of interest are the ELAVL3 and AANAT2, which are associated with the nervous system. The LVAL3 is expressed in neuronal stem cells and developing neurons. And the second of the genes is expressed in the pineal gland involved in rhythmic melatonin secretion. The genes associated with the immune system are the CXCL8B and FOXP3A. The first plays a role in regulating neutrophil migration through the bloodstream, and the FOXP3A gene plays a role in regulatory T cell function. So the materials and methods for RT-PCR are as follows. The genes from primer list that we got were the AANAT2 and CXCL8B, and the novel genes were ELAVL3 and FOXP3A. The RNA samples were collected and run on 1.2% gel agarose with 1x MOPS buffer, and that was used to determine quality of RNA isolated. The concentration of RNA samples obtained using nanodrop 2000C spectrophotometer, and that was at a wavelength of 260 nanometers. The housekeeping gene used was the TATA binding protein, and there were 4 treatment groups: no cDNA, control cDNA, treatment cDNA, replicate 1, and then treatment cDNA, replicate 2. A master mix was also used, which contained oligo-DT primers and Maloney-Murine leukemia virus reverse transcriptase in order to convert mRNA to cDNA. Analysis of the RT-PCR, we used a 1.2% agarose times and 1 times TAE buffer gel with ethidium bromide, and then the gel was imaged using a Bio-Rad imager.
So for the results of the PCR, we had 2 separate gels. The first gel presented the concentrations of each RNA sample. A darker mark on the gel means a high concentration of RNA, which can be seen at the 28S level of the gel, but also less distinct marks can also be seen at the 18S length, which shows that at that point the concentration of RNA was low. The second gel presents the concentration of cDNA samples following the PCR. Again, the bright white spots indicate a greater pixel intensity, which again refers to a greater concentration of DNA. And from the gel, we can see that the only gene that did not work well with the PCR was the FOX3PA gene.
For the discussion, the data from the gene expression was inconclusive in supporting the hypothesis that there were no significant differences in the zebrafish after exposure to 0.5% ethanol. The housekeeping gene exhibited positive amplification. It was concluded that reverse transcription occurs properly, but results were inconclusive for the genes of interest. Previous studies suggested that another zebrafish gene, VANGL2, SHH pathway were influenced by ethanol, creating birth defects similar to FASD, but there are not many studies of these particular genes. FOXP3A did not amplify, perhaps due to the primers not being highly selective or experimental errors during the protocol, and some possible causes of error are failed RT-PCR reaction, inappropriate primers for the targeted gene, or not having an RNase-free environment.
Okay, so the materials and methods for protein expression in immunoblot. A BCA assay was used. There were 2 treatment groups and 1 control group. Samples were loaded into a 96 well plate. The standard used was a bovine serum albumin, or BSA, that was dissolved in an NPB-RIPA lysis buffer. Phage electrophoresis was used. A 10%, 12%, and any percent gels were available. A Western blot, which was used to detect protein in the sample, and then the blot paper gel was used, which also required the blocking buffer and rinse buffer.
So here's an image of the final immunoblot gel, and after consulting with Dr. Havlin, we can say that the results from this gel have been inconclusive.
All right, so the data could not be used to support the hypothesis that gene expression would be downregulated once exposed to the environmental toxin. Control 2 was not visible on the gel because of an error in pipetting. Replicate 1A, 2A, 1B, and 2B all showed up on the gel but led to inconclusive results. This evaluation of gene expression likely did not work because of the pipetting error or due to the fact that the protein was just too weak to be expressed.
So for materials and methods for the enzyme assay, there was a homogenization buffer that was used, which included a 0.05 molar phosphate buffer with a pH of 7.2, 1-time protease inhibitor mix, and a 0.01% Triton X. The standard dilution solution was a 0.1 Na phosphate buffer with a pH of 7.2. There were 2 procedures: glucose 6-phosphate dehydrogenase assay buffer, which is the had a 0.05 molar tris, pH of 7.9, 0.11 mm's of MgC12, and then 0.003 molar G6P, 0.2 NADP plus 1 milligram per milliliter of tetrazolium salt mix. And then the LDH assay buffer was 0.05 molar tris, pH of 7.9, 1 milligram per milliliter tetrazolium salt mix. And these samples were prepared in a 96 well plate and read at 490 nanometers. So to continue on for the materials and methods for the enzyme assay, the G6-PDH had two dilutions, DF1 and DF2. Samples were run in triplicate in a 96 well plate. 100 microliters of G6-PDH assay solution was pipetted into each well, and the plate was then incubated at room temperature for approximately 30 minutes. For the LDH, 3 dilutions. There were DF1, DF5, and DF10. Samples were run in triplicate as well in 96 well plates. 100 microliters of LDH assay solution was pipetted into each well, and the plate was then incubated at room temperature for 5 minutes.
Right, so the results coming from the enzyme assay for the G6-PDH, the concentrations of the enzyme derived from the standard curve were all negative, so no further analyses could be done with that data. But for the LDH, we did have usable enzyme concentrations. However, the DF1 control and triplicate 1 fell outside the curve. So, and the specific activity of LDH was much greater in the control group than it was for both replicate groups, as shown in the top graph. And the same can be said for the LDH units versus the number of larvae in the bottom graph.
The G6-PDH procedure did not yield usable data. This may have been due to the 2 procedures requiring incubation times that were far off from each other. as the G6-PDH required a 30-minute incubation where the LDH required a 5-minute incubation. We also felt that we did not wait the full 30 minutes to incubate, which could have led to negative impacts on the results. And the LDH had a control with a higher specific activity than that of the treatment groups, which suggests that the 0.5% ethanol exposure had a negative impact on enzyme activity of the LDH enzyme. And ethanol has been found to decrease LDH activity in other studies.
So the materials and methods for morphology and behavioral assay, the behavioral analysis was from an overhead view, just to mention. The observing zebrafish after, okay, so the first group was observing zebrafish after exposure to a puff of air. Movement was characterized by directions either left, right, up, down, and the magnitude. Less than 45 degrees was categorized as a slight movement, and greater than 45 degrees was categorized as a large movement. 8 zebrafish from each group were placed into a 24-well plate post-euthanization. For the eye spacing and spine tail curvature, they were scored 1 through 5 based on eye spacing and curvature of tail, 1 was being slight, and 5 being an extreme difference.
So the main takeaway of the behavioral assay was that half of the zebrafish in the experimental groups had absolutely no reaction to the stimulus, which was the puff of air, and all zebrafish in the control group had some kind of reaction. However, there was no noticeable pattern in the exact reaction of the zebrafish. And for the morphological assay, higher scores on both charts represent a lower occurrence of morphological abnormalities. So the control groups on both the spine tail curvature and eye spacing had higher scores compared to the replicates.
The data from the morphological assay suggests that exposure to even low concentrations of ethanol generates changes in development for zebrafish. The target genes mentioned in the hypothesis do not have any role in morphological development of the zebrafish. Therefore, no conclusions can be drawn from this analysis and no relations can be made with the RT-PCR or Western blot analyses. The results of the behavioral assay suggest the same idea. The major difference between the control and experimental groups in this analysis was in the number of zebrafish not responding to the stimuli, and a similar result has been seen by others who conducted an experiment in which they also observed the reaction of zebrafish to physical stimuli. And as stated before, the ELA-VL3 gene plays a role in neuron development, and unresponsiveness could suggest that there is less expression of this gene. However, further analyses will be necessary to further support this claim.
So for any future directions for this study, the main one that we thought of was to have more time for the study, especially for the RT-PCR and immunoblot, because for the RT-PCR, it would allow for multiple primers to be tested for each gene to see which one works the best and which one could actually be used for the study to get the final gel. And for the immunoblot, more time would allow us to have a second gel that might have given us better results so that conclusions could actually be drawn from the data of those two.
And for closing remarks, this study was inconclusive in either supporting or rejecting the hypothesis that Denio Rerio that are exposed to a solution of 0.5% ethanol during development will have no significant change in the expression of the target genes. Limitations of this study include the small sample size of Denio Rerio, limited replicates in each experiment, and inconclusive results from the Western immunoblot. Potential errors could be in researching primers, human error in calculation, or errors in following the experimental protocol. And here are our references.